specializing in neuroscience. I am part of the Crypto Lab in the Department of Human Biology at the University of Cape Town. I am trying to understand why cryptococcal meningitis causes over 100,000 deaths annually. But more specifically, I am investigating whether Cryptococcus neoformans induces hypoxia in the brain. I use the confocal microscopy to visualize and localize the fungal pathogen in the brain and also the hypoxic regions. So by combining immunofluorescence and the 3D reconstruction offered by the confocal, I am able to see a spatiotemporal relationship between the fungus and the host immune cells. That would be from my honors year, where for the first time I saw the mouse brain with a granuloma, which are the lesions caused by the Cryptococcus neoformans. It told me that the science that we are doing correlates with what we see in cryptococcal meningitis patients. Working with frozen tissue causes a lot of background noise. So how I overcame that, I used an optical clearing technique from Light Canvas Technologies, which removes a lot of the background noise and enables me to see a better resolution of the brain. I cannot live without the 20 times objective of the confocal microscopy because it offers me a better resolution and also a wider field of view compared to other objectives, which will enable me to see the entire distribution of hypoxia in the brain. My supervisor is very pedantic about that, so we have established a naming rule that we use. We identify our sample, the stain that we use, the type of markers that we've used, the date and the objective, as well as the type of image you've acquired. And we have a group drive where all of our data is stored, including the metadata and everything, which is backed, hopefully. That's a good one. It would be a psychological thriller. The way cryptococcal meningitis just uses the brain to evade the immune system and just persist is just mind-boggling. And it's why we're investigating cryptococcal meningitis even decades later after its first discovery. Do not break the microscope. But I would advise that you have a clear mind of what you want to achieve before you start using the microscope. Make sure that you have proper training and you have an objective in mind. So acquiring a PhD, obviously. But most importantly, I am hoping to combine all the techniques I'm using and the images I will be acquiring in the confocal microscopy to create a model where we will be able to predict what happens in the brain during cryptococcal meningitis.